Okay, let's unpack this. Think about your body's tissues getting stiff, like a rubber band that's kind of lost its stretch.
Right.
We often see this, things like fibrosis or scarring. Maybe it's just a side effect of damage or disease. But what if that stiffness? What if it's not just sitting there? What if it's actively helping something really devastating like cancer to actually grow? Turns out this physical property, which we might not think about much, is actually a big player in how tumors progress. So today we're doing a deep dive into some really fascinating new research that's figuring out how this happens. And it focuses on these tiny messengers called exosomes.
Yeah, it's incredibly intriguing. We've known for a while, actually, that tissue stiffness, sometimes called extracellular matrix or ECM, stiffening pops up alongside tumor progression. We see lots of cancers, liver, pancreatic, breast cancer, but the exact especially how this stiffness affects the communication between the cells in that whole tumor neighborhood, that's been a bit of a black box. So our mission today is really to break down this quite intricate molecular pathway for you. We want to help you grasp the bigger picture, maybe give you a few aha moments about this really critical but often overlooked part of cancer biology.
Right, so let's start with the basic problem. Tissue fibrosis, that stiffening of the ECM, it's consistently linked to tumors getting worse. We're not just talking about conditions existing side by side with cancer. They seem to actively promote the really aggressive stuff.
Like what exactly?
Well, cells dividing out of control, cancer spreading metastasis, which is terrifying, and even making tumors fight back against chemotherapy.
That's exactly it. And that stiffness, it translates into increased physical, let's call it tension, that the cells can literally feel. They sense these physical cues using special molecules called integrins. Think of them as the cell's little antenna for the physical world around them. Now, we've studied quite a bit about how stiffness triggers signals inside a single cell, but how it affects the chat between different cells in that complicated tumor microenvironment, that's been much less clear, and that's where this new work is so revealing.
Okay, and this is where it gets really interesting, you mentioned, because this study puts these tiny, maybe easily missed players right into the spotlight, exosomes.
Exactly. Exosomes. You can think of them as the cell's own tiny mail packets, little messengers. They're small vesicles, bubbles, really, that cells release. And they're packed with cargo things like signaling proteins, bits of genetic material like microRNAs. They basically deliver messages that can really change what the receiving cell does. It's like a cellular delivery service dropping off specific instructions. And how they're made is interesting, too. They form inside larger sacs within the cell. And then these sacs fuse with the cell surface to release the exosomes out.
And the absolute core finding here, it's pretty stark. Cancer cells grown on a stiff surface, a stiff matrix, they secrete more of these exosomes.
Yeah, a lot more.
It's a direct physical cause and effect.
Right. To figure this out, the researchers did something pretty clever. They grew human liver cancer cells, specifically HA7 cells, on these special gels. These gels were engineered to mimic two different stiffness levels. One was like normal, soft liver tissue, about 0.5 kilopascals. They call that soft. Yeah. The other was much stiffer, like a cirrhotic liver, around 10 kilopascals. That was the stiff one. And what they saw was, well, striking. Significantly more exosomes came from the cells on the stiff matrix. They even called these exhaustive. Way more than from the cells on the soft matrix, the exosoft.
Wow, okay.
And what's really important is it wasn't just a liver cancer thing. They saw the same pattern, more exosomes on stiff surfaces with primary human liver cells, too. And with breast cancer cells, MCF7s, and pancreatic cancer cells, DEC1s, even cells that were precancerous or just starting to become tumor-like showed this increased sensitivity. It suggests that maybe as cells mutate and become cancerous, they get even better at responding to these stiffness cues to send out exosomes.
Okay, so that's the what. Stiff tissues mean more exosomes, but how? What's actually going on inside the cell? How does being on a stiff surface flip the switch to ramp up exosome production? It sounds like a complex chain reaction.
It absolutely is. Yeah. And the study dug deep into that molecular chain reaction. So first off, they found a key signaling pathway called ACT. Gets switched on in cells when they're on that stiff ECM. ACT is like a major switch in the cell. When it's on, it triggers downstream events. And they showed this activation was specific to stiffness. Another pathway, ERK, wasn't affected in the same way.
So ACT is the first responder, in a way.
You could say that. And to prove its role, they did a couple things. They artificially turned ACT on, and boom, exosome secretion went up. Then they used drugs, specific ACT inhibitors like MK2206, to block it. And that significantly reduced exosome release from cells on the stiff matrix. Crucially, they used doses that didn't just kill the cells or stop them growing, showing ACT seems specifically involved in the release part. Blocking it made exosome markers pile up inside the cell like the mail couldn't get out the door.
Okay, so ACT is key for release. Where does the signal to ACT come from? How does the cell know it's on a stiff surface to turn ACT on?
Good question. That brings in another player. Focal adhesion kinase, or FAK. FAK is really central to how cells sense that physical stiffness from the ECM. It's right there at the interface. And they found that if you inhibit FAK, you also reduce ACT activation and subsequently you reduce exosome release. So it shows this neat line. The physical interaction sensed by FAK leads to ACT activation.
So the chain is stiff surface, FAK, activation, exosome release. Is that the whole story?
Almost. There are a couple more crucial links involved in the mechanics of release. Once ACT is active, it influences another molecule called RAB8. Think of RAB8 as maybe a master traffic controller for these vesicles, these packets inside the cell. It helps direct them to the surface for release. The study found stiffness specifically activated RAB8, and importantly, blocking ACT reduced this RAB8 activation. And again, they tested RAB8 directly. If they made cells produce more RAB8, exosome secretion increased. If they reduced RAB8 levels, knocked it down, secretion decreased. It really pins RAB8 as a key player in getting those exosomes out.
Right, so ACT tells RAB8 to get to work. How does ACT do that?
Okay, final piece of this specific puzzle. There's a molecule called rabinate. Rabinate is what's known as a GEF. It's the thing that actually flips the switch on RAB8 to activate it. And the really elegant finding here was that ACT directly modifies rabinate. It chemically tags it, phosphorylates it at a specific spot. This tagging, this phosphorylation, changes rabinate's shape. It forces it from a sort of closed, inactive state into an open, active state. And in that open state, it's much better at activating RAB8.
Wow.
So yeah, the whole chain, to summarize it for you, is like a series of dominoes. The stiff ECM is sensed by FAK. FAK activates ACT. ACT then phosphorylates and activates rabinate. Rabinate then activates RAB8. And active RAB8 drives the increased secretion of exosomes. It's quite a pathway.
That is incredibly detailed. A beautiful molecular cascade triggered just by physical stiffness. Okay, but let's bring it back out. All this technical stuff, FAK, ACT, RAB8, Rebin8, what does it actually mean for a tumor growing in a person, or at least in a model? How does this translate?
Right, the so what question. This is where they moved to in vivo tests using mice to see the real impact. They injected mice with liver cancer cells. Then they treated these mice with either the exhaustive exosomes, the ones from cells grown on stiff surfaces, or the exosoft ones. And the results were pretty dramatic. The exhaustive exosomes significantly boosted tumor growth in those mice. When they looked at those tumors, they had much higher levels of markers that indicate cells are proliferating, dividing rapidly, like Ki67 and PCNA.
So the exosomes themselves collected from that stiff environment are driving growth.
Exactly. And here's the crucial control experiment. They also took exosomes from stiff matrix cells that had been treated with that ACT inhibitor, NK2206. Remember, this blocks the pathway. And those exosomes, they failed to promote tumor growth, which really hammers home that it's this specific ACT-driven pathway leading to those specific exosomes that's responsible for the growth boost.
Okay, that's compelling. Do we know what's in those exhaustive exosomes that makes them promote growth? What's the actual message they're carrying?
Yes, they dug into that too.
Yeah.
And they found something significant. A pathway called the Notch signaling pathway was switched on by the exhaustive exosomes. Notch is, well, pretty notorious in cancer research. It's known to drive cancer progression in many contexts. In connecting this to human patients, they looked at data and found that high Notch activation in liver cancer patients correlated with more severe liver damage and, unfortunately, a poorer prognosis.
Okay, so exhaustive triggers Notch. How's the link?
It seems to be a specific molecule called JAGID1. JAGID1 is a key activator, a ligand for the Notch pathway. They found that cells grown on the stiff matrix produced more JAGID1, but more importantly, JAGID1 was highly enriched inside those exostiff exosomes. So the picture that emerges is that these exosomes, produced because of the stiff environment via that ACT-RAB8 pathway, are loaded up with JAGID1. They travel to other cells, deliver the JAGID1, which then activates Notch signaling in those recipient cells, pushing them towards more aggressive tumor growth.
Wow. So we've gone from just a physical property, stiffness, all the way through this intricate molecular dance with FAK, ACT, Ribinate, Rabidate, leading to these tiny exosome packages carrying a specific pro-cancer signal, JAGID1, which then hits another pathway, Notch, ultimately boosting tumor growth. That's quite a journey.
It really is. It's almost like the tumor microenvironment itself. The physical stiffness is being weaponized by the cancer cells to send out these pro-growth signals.
So if we zoom out, think about the bigger picture, what does this tell us?
Well, it really underscores how much of tumor progression might be driven by mechanisms beyond just direct cell-on-cell contact. These intercellular communications, using exosomes as messengers, seem incredibly powerful. And crucially, they can be profoundly influenced by the physical, mechanical properties of the tissue itself. It raises a big question, doesn't it? How much of what makes cancer progress is actually down to these kinds of subtle signals modulated by the physical environment?
Yeah, it definitely makes you think.
And this research really highlights how vital it is to consider these mechanical factors, not just in cancer, but potentially in understanding how lots of other diseases progress. Think about fibrosis in other organs, or conditions like neurodegenerative diseases or cardiovascular problems where tissue properties change. What other cellular processes, what other signaling might be controlled by mechanics in ways we're only just starting to figure out? That's a really provocative thought here. What else is hiding in the mechanics?